And good morning, micro friends. How's everybody doing today? Woo! Whatever. All right, so it's Wednesday, and I'm still working on exams, and still working on trying to figure out what we're going to do for an article review thing, just for points, but I want to see how the exams go before I really get too worked up over that. Regardless, we're pulling into the next set of chapters 8 and 9, on gene regulation and molecular genetics, which it just so happens is my area of expertise. Isn't that convenient? So you have not had a genetics class, but you've had hopefully some basics of the mechanics of how genes are the coding information to produce proteins. And it's actually a subject that we could spend a lot of time on. We're going to do a little bit of board work and then some PowerPoint stuff. There's a moderate amount of vocabulary, but more importantly, there's some really good, very cool conceptual stuff that hopefully we'll get back to the board a little bit. I kind of like using the board. So a couple of things to think about when we start talking about bacterial genetics as compared to genetics in humans. First off, there is a single circular DNA chromosome that is in the nucleoid, not the nucleus. The reason that's important is the nucleoid is consistent and contiguous with the cytoplasm, whereas in eukaryotes, the chromosomes are in the nucleus, which means that transcription, the production of RNA, is occurring in the nucleus. Translation that is the production of proteins, is only occurring in the cytoplasm. In bacteria, it's all happening in the same compartment, and they actually can coordinate these things, which is very cool. So we've got excuse me, the circular chromosome, as we call it, that's got all the genetic information that the bacteria needs. Additionally, many bacteria have, we can think of as sort of an extra piece of DNA or an optional DNA that is something known as a plasmid. And this is also circular, but it is smaller than the genome. And it has a range of different kinds of genes that might be important for different kinds of environmental conditions. It's also how things like antibiotic resistance can be transferred back and forth. It's an easily transferable piece of DNA. So another thing to think about when we start talking about bacterial genetics is bacteria are, what we say in the biz, they are haploid. That means that they've got a single copy of every gene. So one copy of every gene. Now, in you and I, in eukaryotes, we have two copies. This is referred to as being diploid. You have two copies. Now, what this means is, for the bacteria, if they have a mutation in the gene, they're stuck with it. And you and I, you can have a deleterious mutation in one gene, one from mom, but have a functional gene from the other parent, and you're able to get by. So we can actually exhibit what's known as being heterozygous, and there's all kinds of things that can complicate this. But it's one of the difficulties or one of the complications that you don't have in bacterial genetics is that if you mutate a gene, you mutated it and that's it. They have one copy of every gene. So another part of the bacterial genetics is we've talked about their way they divide by binary fission, meaning they don't produce egg and sperm. So the organisms are clones of each other. They are also asexual. So there is no meiosis or production of gametes. We have another set of terms. I'm going to erase these just because I want these terms to be something we can really hit on. And it has to do with some colloquial terms we use for how DNA and genes can be transferred. There is what's known as vertical transmission. Vertical transmission means that we have the information being passed from the parent to the offspring. And that's pretty much the way genetic information is passed around in you and I. Now, the bacteria and other microorganisms, but bacteria particularly, they also can carry out what's known as horizontal gene transfer. Horizontal transmission or gene transfer, in that they can have DNA passed from one neighbor to the next, particularly in the form of plasmids. So this is not relations, this is just people near you or bacteria near you. So it's neighbor to neighbor. If we think about that from a human perspective, that'd be really weird. We have gene transfer from parent to child, yes, but we don't have gene transfer from our next door neighbors. Probably a good thing. All right. So next, we have what's known as the central dogma. And I always think we should woof whenever we hear the word central dogma. Woof. The central dogma is the flow of information. And the idea of dogma, this is something that is preached a lot. It is the rule. It is how things are. And it's pretty much true. And that DNA can be replicated. And so we'll indicate that with two arrows going back and forth. So DNA can replicate itself to produce more DNA. And then DNA is used as a template to produce RNA. And RNA is a template to generate protein. So this is the central dogma. This is how information is passed back and forth at the molecular level. So not horizontal versus vertical, but when you've got DNA can replicate, and DNA can also be used to generate RNA. RNA is then used to generate protein, and that's the way it goes. You can't skip things. DNA can't be used to directly build a protein molecule. And just as a little further vocabulary, this step is known as replication. This is transcription. And it's referred to as transcription because you're going from one form to another. It's like taking a language and or taking a text and rewriting it. You're transcribing it. When we go from RNA to protein, you're really going from two vastly different macromolecules, nucleic acids to amino acids. This is translation. Now, the last thing to think about is that the source of all variety in all organisms, you and me and bacteria, comes from changes in the DNA sequence, what we call mutations. These are little errors in the genetic code that happened somewhere around there. And this is what gives rise to all the species we have on the planet and all the differences between populations. All right, so here is a great big list of basic terms. And what the heck, let's throw them up and we'll go through them. But it's a fairly easy list. Certainly, these are vocabulary words that you would see on the exam. They're fairly standard. Depending on where you learn your molecular biology out on the street, these are terms that you're going to know, I would hope. Wait for it. Here they come. Boop, 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 boop. So we've got, let me get my little red pointer up. Bing! Yay! Genetics. That is the study of inheritance. The gene is the genetic information for a specific trait. Is the old term. What we know is this trait is a DNA sequence that is actually going to generate a piece of RNA. That is probably the best term, because we can have RNAs that can be translated into proteins. You also have RNAs that are structural, and changes in them can have an effect. So let's just go with the term, a gene is the genetic information to generate a transcript. The genetic material for you and me, that is DNA. And pretty much for all cells and lots of viruses, it's DNA. There are some viruses that are RNA based. We'll get into some virology later, which is really very cool, but there you go. The genome, this is all the genetic information in the cell. So when people talk about the Human Genome Project, that is identifying the sequence of every chromosome. Nucleotides, these are the monomers that generate the polymer. That is the 5-carbon sugar, either the ribose sugar or the deoxyribose sugar, and then the nucleotide nitrogen-containing base that's attached to it. The sequence is a real critical thing because that's the order of the individual nucleotides in the nucleic acids. We also can talk about the amino acid sequence. So just the idea of sequence is the order of these different monomers. Replication, this is the mechanism that is copying the genetic material, whether it's DNA, which is in all cells, or even if it's in viruses in RNA, it's still referred to as replication. Transcription is the term we use when we are generating an RNA transcript from a DNA sequence. So I often think of the DNA sequence as the template, and the RNA sequence is the transcript. Translation, as we just mentioned, this is the process by which we take the information in an RNA molecule and generate an amino acid sequence, or a protein. So the term mutation is really very simple. It is a change in the sequence of nucleotides in the genetic material, in your genome. And so a change in the sequence, there's a couple of things that are interesting about this, is that change from what? Well, you have to sort of arbitrarily decide this is the standard genome. Anything that's changed from this is going to be a mutation. That's one way to think about it. So regardless, though, any nucleotide change is going to be considered a potential mutation. Now, here is some very cool things that we can begin to think about. Back at the turn of the 20th century, so 1900s, Mendel had carried out a whole series of experiments looking at genetic traits and had developed the idea of dominance versus recessive traits, and had begun to establish these rules of inheritance. But what wasn't known was what was, in fact, the inherited material. Probably, if you were back in the 1900s and someone were to ask you how is information being passed from one organism to the next, the best answer might be that it's from protein, because that's the only molecules that at that point were known to actually have interesting structures. Nucleic acid, when you look at it, is about as interesting as cornstarch. It's just a negatively charged polymer. And without sophisticated instruments, you can't tell the ATGC base pairs. So it's like nucleic acid, nah, nothing good there. So a real serious hunt was on for what was the genetic material. And one of the first things that was a really cool experiment done by a guy named Oswald Avery was to explore what was causing a bacteria to actually have virulence. And he referred to this as the transforming factor. And so here is the story. He's got two different strains of a pneumococcus bacteria that will, one will cause a little mouse to die, and one will cause the mouse to not have any bacteria growth and to get up and walk away. So we have the type R, which is non-virulent, and then we have the type S, which is virulent. Now it turns out R is missing kind of the candy coating around the cell, which is what's going to actually serve as a toxin. S has got the candy coating, the peptidoglycan coating, and it is going to be actually quite virulent. So you inject the type S bacteria into a mouse, wait a couple of days, and floop, they flop over dead. Oh, poor little mouse. You rub his belly. All right. So now, a couple of things they did. Avery took the virulent bacteria and heated them up. Killed them, injected, and what happens? Well, the bacteria are dead. You inject it, the mouse survives, and you don't get any bacteria recovered. I'm going to pause for a moment.